Thanks, everyone. So yeah, I'm basically going to be telling you all a bit about the work we've done in Pectobacterium in zucchini. We've noticed that this seems to be an emerging problem in multiple areas. We've seen this sort of crown rot wilting symptom in zucchinis, and yeah, so we've done a bit of work with the bacterial isolates that we've got out of that to try and figure out some basic epidemiology and identification stuff, which will hopefully be, will ideally be the first steps towards getting some good epidemiology for control options and advice. So basically, we wanted to take a look at, first of all, what it actually was, identify the bacteria causing this disease. We want to make sure that the symptoms are something we can replicate consistently and reliably. And we also want to know how it spreads throughout the field. Is cutting your zucchini fruit with tools wounding it, and is it spreading that way? Is it possibly insects? Because we know that some soft rots are vectored by insects, so that could be a possibility. Is it coming through in seed or perpetuating in seed that way? Is it spread through water, crop trash? How well does it survive in the soil? So basically we decided to investigate all of this with some pot trials and field trials, both in Queensland and Western Australia. So first of all, we got our identifications done in WA, thanks to Dom and Craig. So working off of our molecular ID, which is Pectobacterium brasiliensis, for a couple of our isolates, you can see that using these, you can see that using technologies 1 and 2, you do get slightly different identifications. That's more to do with us needing to train those databases better. Basically, the takeaway from this slide is that identifying bacteria can be very difficult sometimes, and definitely more work from a molecular standpoint would be great. These were all recently reclassified as well, so figuring out where they stand in the greater soft rot complex would be definitely worthwhile. So back in Queensland, we started with one pot trial. So we had some potting mix and soil from an infected field, the first infected field that we found here. And basically what we did was grew some zucchinis in that with no inoculum, and in the field and potting mix with Pectobacterium inoculum. So you can see that really we only got very good symptom severity in this one, the potting mix that I inoculated. I tried very hard to kill these. We stab inoculated them. But yeah, for some reason, the field soil, the plants were still pretty happy. My thinking here is that this potting mix is actually not the nicest stuff to grow in. It's a bit sandy and rocky, and maybe that kind of stress was just enough to get us some good symptom severity. But yeah, basically all this tells us is, yes, we can replicate the symptoms pretty well with the method we were using. And is it soil-borne? Not from what we can see here. If you'd think if it was massively, then the field soil should have shown us much worse symptoms. So back over in Western Australia, we have another pot trial happening. Basically, the outcomes of this one was testing similar things. Does contaminated soil that's been directly inoculated result in disease? Not really. So it doesn't look like the soil will retain it particularly well. And does wounding result in disease? Again, not terribly well. So no Pectobacterium was detected in this trial. These are just some photos of what the plants looked like. So back in Queensland, we did a field trial. We wanted to know if tools transfer the inoculum, if inoculating the soil at planting of the seedlings will result in disease, And we also wanted to try our stab inoculum methods again. So this is just a picture of the field, and these dots here are just a layout of the replicates I had. These little red dots are the 2 plants that I inoculated for the cut row. So at fruiting, I'd chop these up and then just go down the row with dirty secateurs as far as I could and see if it spread the disease that way. So the outcomes of this one, interesting and slightly confusing. The tools didn't seem to spread it very well. You would think that if it did, we'd have seen pretty severe symptoms near the ones I cut, sort of tapering off as I went down the row. But I didn't really see that. I got a few randomly infected plants pretty much all throughout the crop. I got Pectobacterium back out of this field. These numbers are a little low due to possibly some false negatives with the direct tissue PCR, but essentially, I got Pectobacterium out of each row, including my controls. So they got both disease and recovered bacteria, which is a concern because we do the controls to make sure that sort of thing doesn't happen. So is the Pectobacterium that I put in the field spreading throughout, or is it happening spontaneously some other way? Back in Western Australia, they looked at zucchini variety susceptibility and also susceptibility in related hosts. We also wanted to know if other Pectobacterium isolates were pathogenic on the related hosts. So this was a ton of work that got some pretty great results out of it. Basically, all zucchini varieties tested are susceptible in some way or another. So you can see here, the B is for browning of the leaves, W is for wilts. You got a lot of wilting in the regal black. And ooze is this sort of oozing from the crown area here. It gets quite mushy and gross, basically. So the different Pectobacterium isolates tested up here, all the Braziliense resulted in some kind of symptoms. Pectobacterium carotavorum seemed to do this as well, but not the oozing symptom. And down here you can see that the Jap pumpkins, butternuts, cucumber, loofah, carrot and tomato did not show any symptoms. But there's plenty that did in the cucurbita and citrullus genuses and in capsicum. So it's definitely worth knowing that and probably investigating this more as well, because that link between what isolates we have and what hosts they infect is a pretty important one. So field trial number 2 back in Queensland, I really wanted to know what was going on with all those random infections. So I want to know if I can track the inoculated bacteria through the field. I also put some insect traps up. I guess, spoiler alert for those, I think this guy's responsible for the destruction of many of them. I caught a lot of mud and mosquitoes, and that was essentially it. So this trial was kind of a disaster in a very interesting way. We had a really prolonged rain period at planting and pretty much through harvesting. This trial did not last long at all. They all went down. They all seemingly got this Pectobacterium disease. And I didn't inoculate the entire field. So this gives me a pretty good scenario in which to track the bacteria that I put in. So I inoculated some of them with a rifampicin mutant. Basically, I can just check in the lab if the thing I put in the field is the thing I'm getting back out. And I could track it pretty reliably. In the plants that I inoculated, I got the mutant back out. In the plants that I didn't inoculate, I did not get that mutant. So that's telling me that there's Pectobacterium coming out of these from somewhere else. And it doesn't look like the inoculated stuff is moving through the field. It looks like naturally occurring disease. And this is within the space of a couple of weeks, honestly. This picture here is your worst case scenario, I think. It went down very fast. So next steps. Determine if it's seed-borne, because all that seemingly naturally occurring disease is kind of suspicious. And that seed I used before wasn't treated, heat treated or anything. So that's definitely something we want to check out. I also want to check out the optimal temperature conditions, because if rainfall, possibly humidity and temperature is a big contributor, figuring out what those exact conditions are will help you probably a lot in planning for when this disease might be an issue. The insect, like my traps didn't work out super well, so I'd probably still look at insect involvement, even if it's just to rule it out. Definitely in Queensland, I think we need to investigate zucchini varieties here and do some host tolerant stuff as well, just to mimic what they did in WA. And we also want to find out if this disease is emerging in other crops and regions, because there is one that does this in Wombok. Basically, Pectobacterium is a pretty wide genus, and a lot of the pathogens within it do have wide host ranges. So that's certainly something important to check out. Recommendations at the moment, all we can really figure out is that risk of infection through soil and crop trash is pretty low, but definitely looking into all this other stuff would help with recommendations a lot more. So thank you to everyone. Thanks to the Western Australia team, Dom and Craig, for doing all the isolate ID and all the trials. Thanks to all the growers and everyone who funded us. And this is a pretty picture of our team. Thank you very much.